Ủa? Hello. Hello. Mm-mm. 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 Kita bagi itu 3 millimeter dengan 5. Dengan 5. 3 millimeter dengan 5 millimeter. Ada. 5. Satu 5. 5. Ah, okay.
Good morning, Dawn. Good evening to you.
Good morning.
Good morning, Dr. Khetarpal.
Good morning, Dr. Vibha. How are you? Good morning, Dr. Homing Jang.
Good morning, Dr. Vibha from Bangladesh. Good morning, Dr. Vibha.
Good morning, Dr. Ketapal and Viva. Do you hear me?
Good morning. Good morning, Dr. Jang. Good morning, Dr. Kwasha Mahmood. Everyone, good morning.
Everyone, including
Dr.
Dr.
Vibha, you are on mute. You are speaking by being on mute.
Good morning, sir. Good morning. I was saying good morning.
Good morning, Dr. Vibha from
Good
Bangladesh.
morning. How are you doing, sir?
I'm fine.
Nice to see you. Good
Okay.
that you could take out time.
Are you fine?
Yes, yes. Doing well, sir.
Thank you for inviting me.
Thank you, sir. There is great interest in this particular webinar, sir.
Yes, we see that.
Number of registrations increased further. So
I see. How many?
have almost 2,400 registered from 66 countries.
Right.
How many countries?
Now 143.
Yes, sir. 140. Still there is time, no, sir? 10 minutes. People will start joining.
Okay.
From how many countries?
66, sir.
56.
66. 66.
Oh,
Double six.
66. Oh, it's great.
Though many people join and sometimes are unable to attend, but still, you know.
Yeah.
Good morning, everybody.
Good morning.
Good morning.
Can my voice be heard clearly?
Yes, sir. Yes, we can hear you clearly.
Okay, great. Thank you.
Sir, Dr. Heidi is having a little difficulty in joining the link. We've just resent her. So we'll wait for a few minutes, 2-3 minutes.
Okay.
Sure. No.
Hi, Fiba. I'm on now. Thank you.
Okay, she has joined,
Okay,
Vibha.
thank you. Thank you so much.
Thank you very much.
Hi, Heidi. Good morning.
Hello. Good morning.
Good afternoon.
Afternoon.
Yeah.
There we go.
Good afternoon.
Good
Thank
afternoon.
you.
Yeah.
Yeah.
Just shut my door.
Vibha, we are dot on time, and I believe we can move ahead. We have
Yes.
about more than 200 people joining.
Yes, sir.
I
So,
think-
Dr. Rishi, we can request Dr. Rishi to start.
Yeah, Dr.
Okay.
Rishi, you are the key player today. Please.
Okay, good morning, good afternoon, good evening, wherever you are. This is the second webinar in the series, what we are organizing on gene editing. And the webinar topic is the advancing genome editing, edited plants from lab to land. And I have pleasure to initiate the proceeding of this webinar by welcoming all the speakers, the chairs, and all the participants. A word about the organizers. There are the three major organizers. One is the APARI, another is the Korea Biosafety Clearinghouse and BCIL. APARI, you know that it was the initiated by Food and Agriculture Organization of United Nations. It is voluntarily member-based and non-profit organization for sustainable agriculture development in Asia and Pacific region. Under its program on APCO app, that's called Asia Pacific Consortium on Agriculture Biotechnology, these webinars are organized. Our valuable partner is the Korea Biosafety Clearinghouse mechanism, which was set up under the Cartagena Protocol, and which is the organization to assist the parties for better compliance of the obligation under the Cartagena Protocol and exchange information about LMOs. BCIL is one organization which is public limited company promoted by the Department of Biotechnology India, and it is a nodal agency to facilitate accelerated commercialization of biotech products and processes. Ladies and gentlemen, these webinars have been guided by a very high-level steering committee consisted of many eminent biotechnologists, regulators, and policymakers. And our today's webinar co-chairs, Dr. Heidi and Dr. Zhang are also the part of the steering committee. So we are thankful that for the steering committee who have been guiding us all through to accomplish this webinar series and later on the resource document, which Dr. Ravi will tell about why we are organizing all these activities. I'm happy to inform to all of you that the interest generated all over the world is tremendous, which is evidenced by the registrants. The participants have registered about 2,400 from 66 countries all over the world. It's my pleasure to introduce Dr. Ravi Khetrapal for his remarks. A word about Ravi Khetrapal. He is the Executive Secretary of Asia Pacific Association of Agriculture Research Institution, that's commonly known as APARI, since 2017. Based at Bangkok, but currently he's working from Melbourne. He is undertaking APARI activities in the region, including execution of the project sponsored by FAO, EU, WTO, ACIR, COA, and USDA on knowledge management, agriculture innovation system, agriculture science technology indicators, pesticide risk mitigation. And currently, he is also serving as chairman of Tropical Agriculture Platform of FAO a G20 initiative for developing and promoting the agriculture innovation system. Dr. Ravi Khetpal, it is a pleasure to have you this morning, and I invite you to present the brief remarks to initiate the proceeding of the webinar. Dr. Ravi Khetpal, please.
Thank you, Dr. Rishi. Well, welcome to everyone once again, and good morning, good evening, good afternoon, wherever you are. Dr. Rishi has given you a brief on what APARI is. And I may just like to add that APARI is working hard as a regional platform to help the Asia and Pacific community to achieve sustainable development goals of United Nations, which you all know where Asia is lacking far behind as per the report from United Nations Economic Social Commission of Asia and Pacific. And we are not even halfway through. COVID-19 has further compounded the situation. We are demographically a very populated continent with 60% of the world population. Having said that, the challenges in agriculture, research, development, and innovation before all of us have become more and more. And some of those challenges are indeed surmountable where the technological innovations are concerned. Though natural calamities and disasters are some areas where we all have to live with and mitigate as much as we can. Coming to the field of agricultural biotechnology directly, there have been challenges all through, right from the investment up to its application and some complications of the uptake of technology. Hostile public backlash in European Union and other parts of the world on GM crops due to ethical considerations from consumers' angle. In this background, gene editing, as you all know, is a very important development that holds promises for all of us to advance and sustain agriculture sustainability and nutrition security. Rich pipeline of gene-edited plants have been developed by both public and private sector across the Asian continent. The application of gene editing is, if you look at the Asian scenario, it needs objective evidence-based information to develop and implement coherent, harmonized policies to guide research. Adoption and trade of gene-edited plants and derived products is a challenge. And in anticipation of this large-scale commercialization, to make it happen in a smooth way for gene-edited plants, there was an important regional consultation held in Korea, the second Asia Forum on Gene Editing, which was held in Gangwon in Korea about 3 years back. Following that, APARI organized the international conference with expert consultation with ICRISAT, and also which resulted in series of recommendations relevant to potential regulation of gene-edited plants. Now, APARI has initiated the exercise of development of a comprehensive resource document on application of gene editing in sustainable agriculture and food security in Asia Pacific region with the support of PCIL and a large number of experts from the region that we hope will provide to governments and other interested stakeholders with information needed to inform policy development, particularly as regard issues related to any potential regulation of gene-edited plants. Regulatory challenges are many, and since the modern biotechnology took birth, we came out from the era of tissue culture, the regulatory challenges have been more and more, and we have seen a lot with the GM plants across the world. Now, the objective of this resource document is to help the countries in Asia Pacific which are at diverse levels of development in terms of biotechnology and its regulation, to have a policy positioning with the government. Unless we have a proper policy positioning in terms of the application of the gene edited plant, we would not be able to have a real commercialization, and we may not have the right kind of investment in biotechnology. For the resource document, we are going to cover all these points. We have developed the outlines of the document already. We will have key issues relevant to research, development, and deployment of gene editing in plant in agriculture. We have identified the lead authors, the well-known players in the field globally, and we, along with our collaborating organization, is hosting a series of webinars to solicit input from key stakeholder groups on the various topics which are covered which will be covered in the document. We will see how best we can diffuse the document once it is developed through a communication plan and by organizing a high-level policy dialogue in the region. However, in the whole process, we have also envisaged having 3 webinars. The first one we had on genome editing tools and its application for targeted plant breeding. We learned a lot through that webinar on the application of gene editing in agriculture in improving food and nutritional security and the various areas that needs to be addressed. We had very useful input from the participants. And in the series, the second webinar, which is going to start now, or where we all are right now, on advancing genome edited plants from lab to land, which is being moderated by Dr. Heidi Mitchell and Dr. Ho-Ming Zhang. We also have in this webinar discussion on case studies from Philippines, Malaysia, and India on the lab to land parameters, issues, and challenges. Challenges from lab to land are plenty. There are technical challenges from innovation to regulation. There are challenges of commercialization. There are challenges of investment in this given field. We had a workshop yesterday only on the impact of such technology on the livelihood and socioeconomic considerations of the smallholder farmers. And we could see that the challenges are multifold and there are means of mitigation of the challenges of investment in agriculture biotechnology, which is the prime or the key factor if we have to push this subject and its application ahead. Now we all need to see how we can overcome many of these challenges through an effective communication. As we learn, the science communication part is something which is lagging behind and not keeping pace with the research which has come up. And also we need to have an aggressive and a focused knowledge management and capacity building programs. Well, with these words, I would like to sum up by profusely acknowledging the support of Korean Biosafety Clearinghouse, support both technical and financial, and of BCIL for collaborating actively with APARI, and also to all the participants who are involved in this webinar. Special thanks to all the esteemed members of our steering committee, which Dr. Rishi was mentioning, who carved the roadmap of webinar series for initiating the exercise of reviewing the genome editing tools, its application from lab to land, and enabling policy positioning, which in fact, this enabling policy positioning will be the next in the series. Thank you, one and all. I wish you all useful deliberations. Thank you.
Thank you. Thank you, Dr. Ravi. And without taking much time, let me invite the co-chairs or moderators of this webinar, and I'll take an opportunity to introduce very briefly. First, Dr. Heidi Michel. Dr. Heidi Michel is the Director of Contained Dealing Evaluation Section at the Office of Gene Technology Regulator in Australia. Heidi joined the OGTR in 2006 as an evaluator preparing risk assessment for a range of GMOs. She has also worked in the regulatory practice section of the OGTR at interface between scientific risk assessment and operational regulatory policy. She has represented Australia at OECD and CBD Sabesta meetings and presented talks at domestic as well as international conference on regulation of GMOs in Australia. Heidi has a PhD in plant biochemistry and worked as a research scientist for 10 years in the UK and Australia, publishing papers in peer-reviewed journals, including Nature. Welcome, Dr. Heidi, to co-chair this session. I invite you, and I'll take a minute again to introduce our another co-chair, Dr. Ho Min Jang. Dr. Jang retired in 2018, now an emeritus researcher at Korea Research Institute of Biosciences and Biotechnology, after he had retained the position of KBCH, that is Korea Biosafety Clearinghouse Director, for 15 years, still acting as a national focal point of BCH. He has engaged in biosafety for over 20 years. He took part in biosafety working group in drafting the Cartagena Protocol on Biosafety as a national delegation in the late 90s and early 2000. Sorry. Internally, he has led the way, established the national biosafety framework from setting up to KBCH to facilitating the institutional functioning of the national framework. He consistently emphasized the importance of regional cooperative activities in the last recent COP meetings, while echoed by several participants. His efforts actually paid off in a way to concept of the regional cooperation being integrated into decision papers of practical implementation in several articles of the protocol in the COP 10 in 2018 and the previous one in 2016. With these words, I want to inform to all of you that these two co-chairs are also our steering committee members. They have guided us and advised how to develop this program in the webinar series and ultimately culminating into a document. So Dr. Heidi, Dr. Jang, I invite you, please co-chair this, and the screen is yours. Please conduct the proceeding of the webinar. Thank you so much.
So good morning. Thank you for the very kind introduction. I think Dr. Jang and I are very excited to be chairing this webinar today. As was mentioned, Dr. Ravi Katapa mentioned, the previous webinar discussed the tools and the applications for targeted plant breeding. And we heard a lot about different examples of where targeted plant breeding or gene editing had been used in the lab. And also, I think Dr. Botello talked about other advanced gene editing tools that could be used in the future. So today's webinar is going to move on to getting those products, the gene edited plants from the lab and out into agriculture. So we've got 3 speakers this morning. We have Dr. Don McKenzie, who's talking about the context and status for regulation of genome editing in agriculture. We're then followed by Dr. Peter Thiggeson, who's talking about how the OECD views genome editing, and he'll also talk about Australia's perspective. And thirdly, we have Dr. Florida Carino, who's talking about the conventional and biological diversity and the Cartagena Protocol and the views that she's seen expressed there on genome editing. We then have a panel discussion with 3 speakers who are going to talk about country perspectives for genome editing. So I'd like to hand over now to my co-chair, Dr. Zhang.
Dr. Zhang, you are muted. To unmute.
Okay. Sorry for that. Thank you, Dr. Ketapal and Dr. Yagi, and my chair. First of all, I would like to say that I'm impressed by what Dr. Ketapal mentioned during his speech regarding the context of this magnificent event being organized. That is, this event has started from the second Asia Forum on Biosafety with the theme of gene editing held in 2018 Gangneung, Korea, which I organized and hosted. My primary intention at the time was to provide an opportunity for government officers of Korea to have access to the worldwide landscape and detailed information surrounding gene editing, ultimately to help them streamline the relevant national policy within the right time. However, to conclude, I only get to ask myself if my efforts with such intention was really paid off enough, thinking Korea is still likely to need some more time to overcome some difficulties before the regulatory policy on gene editing is put in place. Instead, I am relieved and even pleased with the vivid memory of the vibe and willingness to engage in that the invited eminent speakers and participants had shown through to the end in the discussion. And I remember that as that event came close to the end, we found ourselves researching the keyword that could explain the reason why people came together to that remote place from the world over. Of course, people from Asia included. I dare to say we finally found the key word. It was context that we shared each other in the ending part of the final day. We went to the point that whatever different policies or decisions countries had taken or will do regarding gene editing, the context behind would be the keyword that we have to bear in mind to understand different institutional establishments in countries. That is, the story would be like this. Although we did not speak it out explicitly, the better we get to understand the contexts in countries in making their gene editing policy, the more valuable lessons will we get from each other. And this would help us come closer to harmonization by overcoming whatever differences among us. In this sense, I would like to believe this series of webinars on genome editing will surely contribute to better understanding the contexts behind the status quo in countries. I once again thank the organizers for making alive and connect the Gangneung event to the subsequent steps to this big event. I know Dr. Viva Ayuza and Dr. Morgan McLean took the initiative for that. I appreciate very much. Yeah, this is ratings from my side as a co-chair. Then now, shall we open the second one of our webinar series? First, let me introduce the first speaker. Actually, we have I think we have a very brilliant lineup of esteemed speakers and panelists. Yeah. The first speaker is Dr. Donald J. McKenzie. He is the Executive Director of the Institute for International Crop Improvement. The acronym is pronounced as Ilse or Ilse? Ilse? Yeah, I think Ilse. Yeah. At the Donald Danforth Plant Science Center. He manages the ILSE's program and partnerships dedicated to delivering precision genetics faced by genetics technologies to meet the most significant food and nutritional security challenges faced by smallholder farmers everywhere. And he has served in various leadership roles in the Canadian federal government, the agriculture biotechnology industry, and in public sector research and development organizations. Well, shall we welcome Dr. McKenzie?
Thank you, Dr. Jiang, and good morning, good afternoon, and good evening to all. Thank you as well to APARI, to the Korea Biosafety Clearinghouse, and to BCIL for the invitation to participate in this important dialogue on advancing genome-edited plants from the lab to the land. And lab to land really encapsulates what our mission is at the Institute for International Crop Improvement at the Danforth Center. We are really focused on trying to close the gap between innovation and impact. And all of our work is done in collaboration with National Agricultural Research Institutes and other international institutes around the world. And so none of the work that we do is done alone. It's always together in a large team. And our role in many of these instances is not just in provision of technology, but in helping to carry out the translational research and to provide the regulatory support that's often needed to get these products actually into the hands of farmers. So the past few weeks have seen some really highly significant developments for public sector research and being able to deliver products of biotechnology either directly into the hands of farmers or at least getting almost there. So we've seen the environmental release of virus-resistant cassava in Kenya, only the second genetically engineered food crop ever approved for environmental release in Kenya, and that happened in the middle of June. We have the first seed sales, the commercial release of pod borer resistant cowpea in Nigeria. This represents the very first wholly public sector developed genetically engineered food crop released to farmers in Africa. And finally, we have golden rice, which was approved for commercial cultivation in the Philippines. And there are a couple more that aren't on this slide that are equally important. And those are the food approval of BT eggplant or BT talong in the Philippines, also on the 21st of July. And then even more recently, Indonesia has registered the first potato cultivars that contain bioengineered resistance to late blight disease, Phytophthora infestans, the causal agent of the Irish potato famine. So each of these achievements is noteworthy in its own right, but I think the other realization we have to come to is that at least for the three that are shown here, they represent technologies that are anywhere from 15 to 30 years old. The first description or publications on the use of RNAi technology for virus resistance came out in the early 2000s. The BT technology that's in the pod borer resistant cowpea was first commercialized in the United States in maize in 1996. And then Ingo Petrakis and Peter Beyer described the first events of golden rice back in the late 1990s. So the point that's worth making here is that the gap that's between innovation and impact for these products is really large. And we have to, I think, collectively work to urgently and significantly reduce these gaps. If we think that new plant breeding technologies or new plant breeding innovations are going to get meaningful contribution to achieving sustainable development goals. So although the focus here is on how to manage or how to regulate or not regulate products of genome editing, I think it's worthwhile going back and just revisiting, well, how did we deal with the last new technology in plant breeding innovation? And so this would be roughly about 30 years ago, around about the time when some of the products I just described were being developed. And back then, it was really quite reasonable to ask the question whether or not the products of this new genetic engineering technology presented new hazards or risks, and whether the products or the technology itself should be regulated, and then how or if to do so. Back at what I'll call the beginning, around 1987 or so, for plant genetic engineering, at least in terms of thinking about potential commercialization, we had learned scientific societies from many countries come to the opinion that, you know, no unique hazards existed. The risks posed by products of genetic engineering were really of the same kind as those associated with more conventional forms of breeding. And that risk really was not related at all to the process used to create the product, but really related to the product characteristics, to the phenotype of the plant, if you will. But notwithstanding that advice from scientific societies, and that every country that wanted to either produce genetically engineered plants or make use of those products in agriculture or in commerce went down the road of creating biotechnology or biosafety regulatory frameworks to specifically manage those products. And we know where we are now. We are in a situation where these products are amongst the most highly regulated anywhere. The time and cost to move from innovation to impact has become extraordinarily high. And largely it's an endeavor that can really only be done successfully and routinely by large transnational corporations that have all of the resources necessary to do it. Information and data requirements have escalated, and we've got the complex situations around international trade and the low-level presence that we have to worry about for products that might be approved in an exporting country but not in an importing country. And we really have to, I think, ask the question sometimes whether the regulations and what we wanted them to achieve have actually been achieved. And for those of us working in the public sector who don't have infinite resources, we're often also working in geographies, whether it's in Asia or in sub-Saharan Africa, where many times the regulatory pathways are not yet well developed. And there's an absence of clear guidance, and there are weaknesses either in capacity for risk assessment or in capacity for enforcement, if you will. And oftentimes the products that we're working on, such as a Bt cowpea or a virus-resistant cassava, that are amongst the first ever to be looked at and reviewed by a regulatory agency. And that always becomes a bit problematic. So now we're at this stage of, well, how do we deal with genome editing? And in the first webinar, you heard and learned of all the exciting potential products that could be coming out of this new technology. So we'll not talk about that here today. But I think it's worth pointing out that conversations about how genome editing and plant breeding innovation should be regulated or not need to be rooted in the historical context of plant mutagenesis tools. So there was this nice graphic in a publication by Shalaki et al. Of a couple of years ago, where they were looking at the evolution of plant mutagenesis tools. And I thought it quite nicely captured where we've been and where we perhaps are at the moment. And we're starting, back in the early part of the 20th century with the first use of radiation, X-rays, if you will, to mutagenize plants. And that was used to create new varieties of maize and barley. And that was described back in 1928. And that was followed in the mid 1940s by use of mutagenic chemicals to induce mutations in many crop species. And just using those two techniques, the International Atomic Energy Commission has a database of mutant plant varieties, which by today contains more than 3,350 varieties of mutated crop plants that represent more than 210 different plant species. Over time, these tools of mutagenesis have evolved. They've included more precise molecular techniques, such as zinc finger nucleases, talons, CRISPR-Cas, and that. And the theme throughout all of this is that we're moving over time from undirected, if you will, untargeted techniques for introducing mutations to techniques that are very precise in where mutations can be introduced. All plant breeding methods give rise to unintended effects. And, mutation breeding using either radiation or mutagenic chemicals, all of the effects are off-target effects. There are no targeted effects. And yet, even with this technology, we find that the conventional agri-food regulatory system is quite sufficient to provide the necessary oversight for products from mutation breeding. We're generally comfortable with having these products regulated as conventional. So why should we treat targeted mutation products, such as products from genome editing, from CRISPR-Cas, from prime editing or base editing, any differently than we would treat products of radiation or chemical mutation? And just a brief reminder of what does the conventional agri-food regulatory framework look like? And of course, it goes all the way from crop improvement, regulating how new varieties are evaluated and assessed, ensuring that they are both safe and efficacious in terms of demonstrating value for cultivation and use for farmers. We've got systems in place to ensure that quality seed is provided to farmers through certification schemes and that. We have rules and regulations that govern, in many cases, quite precisely how crops can be cultivated and grown, whether it's regulating the kinds of inputs from herbicides or insecticides that can be applied, or ensuring that pesticide residue levels are within tolerances so they don't pose any safety hazards to consumers, all the way through to food manufacturing, food sales, and distribution. Typically, these involve multiple agencies within the government that encompass agriculture, environment, pesticide registration, public health, international trade, and so on. And then our international standard setting bodies that are involved at different parts along the chain as well. So this sort of umbrella set of regulations and legislation has done a really good job of ensuring a safe and nutritious food supply for consumers over many decades. And so I think we need to think about how this collection of conventional regulations can apply to genome edited plants and their products, and whether or not it's going to be sufficient or whether we need anything extra. There are, I think by now, 173 countries that are parties to the Cartagena Protocol on Biosafety. And for those countries that have been basing their domestic legislation and regulation on the definition of a living modified organism within the protocol, they've decided to take advantage of or use the fact that an LMO means something that possesses a novel combination of genetic material. And many countries have taken the position that when we're talking about, for example, simple knockout mutations, SDN1 type of products that you, I'm sure, heard about in the first webinar series, and even SDN2 products that contain perhaps small changes or additions to the genome, that these in fact do not represent novel combinations of genetic material. And so many countries would take the view that these products are not LMOs and would not fall under the regulatory remit of their existing LMO regulatory framework. I'm going to give one example, and this is the example from Nigeria, which just this year finalized its national guidelines for the regulation of gene editing. Within the guidelines, they provide a very nice review of the state of play of genome editing technology, and they make note of the definition of an LMO within the Cartagena Protocol, and are basically taking the approach that gene-edited products that do not contain introduced transgenes or DNA will not be regulated as LMOs. And so that would include SDN1 and SDN2 products once they've been shown to be free of any introduced DNA. And on the one side of this chart, we have the schematic of the flow chart for how they would deal with applications of genome editing. And it's really about whether or not the product contains new introduced DNA or not, as to whether it falls under the remit of the GM legislation. Looking around the world, this is a summary chart that I adapted from CropLife International. What it does show is that, at least within the Western Hemisphere, there's a growing consensus, if you will, on how different countries plan on treating gene-edited products, with many of them deciding that products that don't contain introduced DNA or transgenes will not be regulated as if they were LMOs or GMOs. The rest of the world, though, is looking quite different at the moment. We have Europe and perhaps New Zealand that stand a bit apart. We have a number of countries that are still in the process of deciding how they're going to deal with gene-edited plants. And then we have some countries where perhaps the discussion has not even started yet, and it has yet to get going. What this chart really doesn't show very well, though, is some of the nuances on how different countries are approaching the problem. So, some of them, such as Australia, for example, are taking a differential approach as to whether the product is classified as SDN1 or SDN2. When you look at the United States as another example, there's not yet total coherence across the different regulatory agencies. And you don't appreciate that even the exemptions that were provided under the new secure rule promulgated by the US Department of Agriculture earlier this year, is that those exemptions, they don't apply to polyploid plants. So for example, the exemptions that would exist for maize, for example, and an STN1 mutation there don't apply to potato or to teff or to canola. And Canada, of course, is also unique amongst all of the countries in that ever since 1993, Canada has taken a product-based approach to regulation that really doesn't look at the process. So products of conventional breeding, products of mutation breeding, products of genetic engineering, or even products of genome edited are all captured under the novel plant and novel food regulatory regime in Canada. So they don't have to worry about changing their legislation or regulation every time the technology changes. The one difficulty with this current patchwork approach, of course, is that when we think about international trade and the movement of seed or grain or even food around the world, that in order to facilitate trade and make sure that we don't have barriers erected, it's really important that countries take a consistent approach to how they're regulating agricultural commodities, including products of genome editing and that. In the existing regimes around genetic engineering, even though countries have taken different approaches, they generally have all decided, well, they're going to regulate it one way or another. And here with genome editing, I think that the hope and the desire is that countries move towards creating sort of exemptions that will allow these products to the marketplace sooner. So just some final closing comments, and to make note of, the vast majority of the market-oriented plant breeding innovations that we're talking about are mainly what we would call SDN1 and SDN2 products, and are virtually indistinguishable from the kinds of mutations that you theoretically could achieve using more classical methods. Regulatory systems ideally should be, obviously risk-based, but not tied to any breeding technology. I mean, we've talked about, right now, we're talking about genome editing, but in the future, what about epigenetics? And what about, using changing methylation patterns to result in different plant phenotypes? How are we going to regulate those? If we keep on linking our regulatory frameworks to the technology of the day, we're always going to be behind. We'll never be catching up. And so we need to focus our regulatory frameworks more on the characteristics of the final product, not the way it was produced. Creating an enabling policy environment, we really need to do that for our local researchers in that, for homegrown solutions to meet local food and nutrition security problems and that. And the real winners here will be smallholder farmers at the end of the day. The challenges are great. So we know Sustainable Development Goal number 2 is to eradicate hunger and achieve nutrition and food security, as well as sustainable agriculture by 2030. However, we also know that the statistics are going in the wrong direction. Hunger actually has been increasing, and it has been since 2015, and it's only gotten worse as a consequence of the recent COVID-19 pandemic. We only have 9 harvests left between now and 2030. So we really do need to think about how we can shrink that gap between innovation and being able to provide safe technology and impactful products for smallholder farmers. Thank you very much.
Thank you. Thank you, Dr. McKenzie. I think you gave us a very elaborate introduction on many issues, from trying to define the definition of the products edited by genome editing, to practical issues, to such as trade. And yeah, so I think many questions would be waiting for you to answer, but I think it a little bit behind the schedule. So I would like those to be left until after the panel discussion time. Okay, then I would like to turn over this mic to my co-chair for the next. Hi, Heidi. Yeah.
Thank you, Dr. Zhang.
Yeah.
So I'd also like to thank Dr. McKenzie for a lovely talk. Our next speaker is my colleague, Dr. Peter Thigerson. He's the Principal Regulatory Scientist at Australia's Office of the Gene Technology Regulator. He's also the current chair of the OECD Working Group on Regulatory Oversight of Biotechnology, harmonisation of regulatory. Peter, you'll have to introduce that one. The focus of his role at the OGTR is risk assessment of GMOs, and he's interested in the implications of new technologies and other biological assessment frameworks. He has evaluated environmental release applications for GM plants and also has experience with legal and regulatory policy issues. So Peter, I'll hand over to you now. Thank you.
Thanks very much, Heidi. Just unmuting myself and I'll see if I can share my screen. And launch that to there. So hopefully you can all see that. Now I'll just turn the laser pointer on. So Heidi's introduced what I'm going to talk about. Good morning, good afternoon, good evening, everybody. Thank you very much to the organisers for the opportunity to present as part of this webinar. So this is an overview of what I hope to speak about, or for you to take away today. I'm going to probably go over some of the ground that Don has just gone over, but maybe from a different perspective. So some of the background to genome editing and its regulation, and trying to frame that as what's the problem. I'm going to spend some time talking about definitions and principles, and the current Australian regulation will provide something of an example for that. And I will also talk about OECD activities. So as Heidi said, I'm the current chair of the Working Party on the Harmonisation of Regulatory Oversight in Biotechnology. So it is a bit of a mouthful. The very simple key message that I hope everybody takes away from this talk is to have clearly in mind the difference between legal and scientific definitions and legal and scientific arguments. And Don spoke about enabling policy environment, and I'll come back to that. And a disclaimer, this is my own analysis. This isn't the view of the Office of the Gene Technology Regulator, nor is it the view of the OECD. The other thing I wanted to say, I actually wrote out. So noting the introductory comments, I'm going to give you some observations about issues and actors who will ultimately determine what the regulatory outcomes for the application of genome editing in plants are. Or to put that very simply, you have to consider who will decide what those outcomes will be. So Don also already touched on this history, and I'll reiterate very quickly on this slide. I won't go through everything. So recombinant DNA started in 1975. There was a conference in Asilomar where the scientists adopted what we would now call a precautionary approach, where they were uncertain about what this stuff of life mucking about with it would do. In about the year 2000, individual jurisdictions and internationally with the Cartagena Protocol on biosafety, laws are instigated by jurisdictions to regulate this new technology as it was coming to market. In fact, coming to market before then in the mid-1990s. If we jump ahead through new plant breeding technologies to genome editing, and I'll just mention synthetic biology, it's possible that the speaker following me will talk about that in the context of the Cartagena Protocol. But probably the main thing I want to iterate here is we have some legal rulings on whether gene-edited organisms will be subject to GMO regulations, and principally the one that everyone I think is aware of is the European Court of Justice ruling in 2018. So some context. So I said around the year 2000, we have recombinant DNA or GMO laws introduced. It was a new technology, a precautionary approach with a requirement for pre-market safety assessments, but with the intentional exclusion in most of those systems of what we might call traditional breeding. And Don went through in a nice detail the mutagenesis techniques. So kind of three different approaches adopted globally to regulating recombinant DNA. One was to have a process trigger, just kind of in line with that Asilomar beginning, and virtually all the countries of the world have adopted this approach, and most of the Cartagena Protocol signatories have this approach of a process trigger. The EU does, Australia does, Korea does, a number of the other countries present in today's participants do. The second approach is a product trigger based around novelty. Canada and New Zealand, to varying degrees, have such a trigger. They also consider the process, if you look behind how they actually do their assessments once the capture for regulation has happened. And the third approach is to adapt existing laws without making any GM specific laws or laws which are intended specifically to capture GM. And the USA is the best example of this, but that also considers aspects of product and process, and the adaptation of the US plant health laws, I think, leave it off the early technology of agrobacterium transformation, where agrobacterium is considered a plant pest. Using Australia as an illustration, we have a process trigger. We have very broad definitions, but with some exclusions. So a very broad definition of a GMO, organism modified by gene technology. And gene technology is also very broad, any technique for the modification of genes, but with some exclusions. And as an example, radiation and chemical mutagenesis were excluded from the techniques that constituted gene technology for the purpose of regulation. So some of the international context, Don has also touched on this, but basically have different countries. Many of them have different laws and legal systems. For their GMO or biotech laws, they've adopted different definitions, and they also have different approaches, different policies, and different publics, and that can all lead to different regulatory outcomes. So it leaves the question of what is regulated and how is it regulated in each of those jurisdictions. I'll just jump over this slide quickly. Don's also touched on this a little bit. If you consider the classical GMOs, a small number of species and a small number of traits with a small number of large players in the development side. For gene editing, the technology is more tractable. We've got a large number of species under research, a large number of traits, and a lot more smaller entities that are using this technology to take it forward. So what does the problem look like? Circa 2000, what everyone was regulating or setting up their regulation for was the classical transgenic, with a promoter from one species, a gene from another species, a terminator from another species, and irrespective of the definition, everyone said that's a GMO. We jump forward to 2021 with genome editing, with possibly single base pair substitutions with the same definitions by and large that operated in 2000. The question is, subject to those definitions, are the plants or animals or microbes with these types of modifications or genetic changes genetically modified organisms? This is a busy slide. I'm conscious of time, so I'm concentrating on this stream down here. And this relates to the comment I made at the beginning about who are the actors. So governments make laws, they define what's to be regulated. You might say, if you want to have a discourse about the policy settings, it's governments, not regulators, that you should be talking to. Regulators administer the laws as given to them by government, and that's where science-based regulation comes in. And in many jurisdictions, the decision, if there's a dispute about whether something is regulated, will be decided by a court, not by the regulator and not by the government. Touch on this one very briefly as a preamble to the next slide. If you're designing your regulation for whatever it is, consideration of what your protection goals are, what's the information underpinning the legislation, what's the risk, what's the science behind it, and also societal values go into making the law. But once the law is made, all of those other stakeholder considerations, if you like, I think disappear. So this is our view as a regulator in Australia, that one cannot interpret laws to say, this is what it meant to say, or this is what it should have said, or from speaking to a lot of scientists, well, the terms of art have changed and that's what it should mean now. No, it's fixed in the law in the way it was set by the government at the time. So what are the reactions? I've mentioned the European Court of Justice. Don has also mentioned New Zealand. They had a case which went somewhat unremarked as far back as 2014. And the conclusion of both of those cases, and so for both of those jurisdictions, is that if your plant or animal is genome edited, it's a GMO. In other countries, the process is ongoing. Argentina and Brazil have kind of taken a novel way where they do a pre-assessment, which includes legal and some scientific aspects, as I understand it, which will decide whether the organism is to be regulated. I'll touch on Australia some more as we go. Canada recently had a consultation on some guidance, which was framed much more in the context of keeping track of genome edited organisms, which might not necessarily have had to go through a regulatory process. And Don alluded to the US secure rule. So that's just a short grab bag of some countries' approaches currently. The Australian system, there are two sets of definitions, one for GMOs and one for food, a process trigger, as I've said, and that feeds into science-based case-by-case assessments and approvals. This slide, also very busy, but there have been a large number of scientific journal publications over the last 2 years that have spoken about the regulation of genome editing. And once again, this is my theme of terms of art in science versus legal definitions, and that the words matter. So when I first saw this, one of these nice graphics, it says that and Australia is given as green, genome edited organisms are not regulated as GMOs, and if there's no foreign DNA. unfortunately, that's not actually quite correct. Some genome edited crops are not regulated as GMOs in Australia, whereas some are regulated. So it's like, from a scientific perspective, going back to the original paper rather than going on someone else's gloss of what the original paper said. Get all of these things to come on board. Go back one. The very short story about what is excluded under Australian regulation for genome editing is SDN1 modifications where no guide template for repair is added. So that's what's excluded. Whereas what is termed SDN2, an oligo-directed mutagenesis, where a template for guiding repair is added, they are considered to be GMOs. In terms of the Big P policy situation, there was a review of the whole Australian GMO scheme starting back in 2017, but very simply, I'll say the work is ongoing there. No specific amendments were proposed by that review, but we may see something come out in the course of the next year. Similarly, in the Australian food space, a review has been being undertaken by our colleagues at Food Standards Australia, New Zealand. Nothing has been published on that. That work is ongoing. So once again, watch this space. Okay, jumping to OECD. So said, I've participated in this working group, which is really about environmental biosafety, which you can say historically has been about transgenic organisms or GMOs. Its major output is consensus biology documents to serve as the baseline comparator information where assessments are required. There's a sister group for foods and feeds, and they produce compositional documents, which is related to substantial equivalence. And the idea is to have a common base of scientific information to inform assessments. I'll just show this slide very quickly, the OECD principles from very, very early on. If you have to do an assessment, an environmental assessment of an organism, whether you call it a GMO or not, it's a trait-based assessment suggestion. So the parent species, the receiving environment, and the trait contribute to your assessment. In terms of genome editing specifically, in 2014, so questions were being raised about whether, with the advent of what were then called simply new plant breeding techniques, of whether current approaches and paradigms for environmental risk assessment of transgenic plants were applicable. And so a workshop was held, and the very short story conclusion, and I'll read the whole thing out, is that current guidance and tools for environmental risk assessment of transgenic plants are applicable to plants developed by new plant breeding technologies where such an environmental risk assessment is required. Closer to where we are today, in 2018, OECD hosted a conference about genome editing and its implications for health, environment, and regulation from agricultural applications. Touched on three elements: the technical developments, risk assessment issues, and I think relevant to our discussions today, regulatory and policy approaches in various countries. The proceedings of the whole conference were published in a special supplement in transgenic research, so you can see what each of the countries that presented, particularly in the regulatory policy space, had to say there. Currently, and since 2014, information sharing became, for what I would call genome editing, became a standing item on the agenda. And the take home here for the audience to see what other countries are doing is that from this year, what's called the tour de table, this information sharing, including relating to NPBTs or genome editing, has been published. So that's the first time these country reports are published, and you can find them at that web link. So I'm going to come in just on the time I was given. To recap, Don said this, very rapid scientific advances, genome editing potentially being applied to many species in many environments with many traits. A key part of the debate is about definitions and country policy approaches. Some of the principles underpinning this debate is the legislation that we started with was set up with this precautionary approach. I've touched a little bit on the global state of play and its implications. And there is an ongoing regulatory policy debate. And my take-home message, particularly for the scientists, and recalling Don's comment about people who are working on this wanting to influence the policy environment, is to concentrate or very clearly clarify in your mind what's the scientific argument, what's the legal argument, and to recall who are the actors that determine how the regulation is set, and to dialogue with those and not to pick the wrong actor to dialogue with. So I'll stop there. Thank you very much.
Thank you very much, Peter, for that really interesting and clear talk on who we should do if we want to make change.
Have I managed to unshare my screen? No, stop share.
No.
There we go. Sorry. Okay.
Brilliant. Thank you very much. So given the time, I think we'll save the questions till the end when we've got the panel discussion. So if anybody has questions for Peter, please could they put them into the chat. I'd now like to hand back to my co-chair to introduce the next speaker. Thank you.
Thank you, Heidi. And the next speaker is Dr. Florida Carino. She retired as a full professor of biochemistry from the University of the Philippines, Diliman, and she was a former director of 2 institutes of the university. Dr. Carino is currently the physical scientist member of the Department of Science and Technology, DOST, Biosafety Committee, and has been involved in drafting policy and regulation of genetically modified organisms since 1994. Dr. Carino, please take this microphone.
Yes, thank you. I would like to share my screen. And let's see if I can find it. So I'd like to start from scratch. Okay, so you had the last slide shown first before I started. I decided just to show that one, mainly because that was a comment that has been made by one of my colleagues who's starting to work on genome editing. She said that for every innovation, there seems to be a regulation. And then after that, an international convention that can drive my innovation into oblivion. And sometimes when I attend the meetings of the CBD, the CPB, and discuss genome editing, sometimes I have the feeling that she probably is right. Anyway, before we go through the different alphabets of genome editing, I would like to have a first comment that I would make with respect to the CBD and the CPB. It is seldom that you're going to see the words genome editing or gene editing outside the brackets in any of the conventions on modern biotechnology that you'll find, both in the Convention on Biological Diversity and on the Cartagena Protocol on Biosafety. So the major topic of our talk, genome editing, is not even usually said together in any of the conventions that we have actually agreed with. Okay, so Agenda 21 actually commits all the states towards sustainable development. And of course, it looks at every area of human activity that would impact the environment. So to be able to come up with a mechanism for translating the principles of Agenda 21 into practical terms, we actually came up with the Convention on Biological Diversity, CBD, the first 3 letters for an alphabet, whose three major goals would include the conservation of biological diversity, the sustainable use of its components, and the fair and equitable sharing of the benefits arising from the utilization of these genetic resources. And they cover everything. So this international convention has 196 parties. Okay, and so if you look at the world map, you'll see that only 4 countries are not members of the Cartagena of the Convention on Biological Diversity. The US is a signatory, but it has not ratified this treaty. Then you have a few others. We have, for example, other non-members will include Iraq, Andorra, and Somalia. The CBD implements its goals through 2 protocols and 1 supplementary protocol. The Cartagena Protocol on Biosafety, so if we rumble the terms a little bit, it's CPB, with its sub-protocol on liability and redress, known as the Nagoya Kuala Lumpur sub-protocol. And then for access and benefit sharing, we have the Nagoya Protocol on access and benefit sharing that will try to share the benefits that may be derivable from the genetic resources of one particular party. Okay, so initially discussions on developments of technology was actually brought to light in the COP-MOP in Bonn, Germany. COP means Conference of Parties, MOP means Meeting of Parties. So when we're talking about Cartagena Protocol on Biosafety, we will be usually using the terms COP-MOP. But for the Conference of Parties that usually just deals with the Convention on Biological Diversity, we will usually just say COP. So this particular COP tried to articulate the mechanism or the procedure for identifying new and emerging issues so that the discussions, or shall we say the topics that need to be discussed in the COP would not be too many, and would also mean that you do not clutter the agenda with too many things that you have to consider in a limited amount of time. So the SynBio, if it happens to be a new and emerging issue, would have to satisfy the criteria that has been set in decision COP9, item 29, paragraphs 11 and 12. The NEI criteria, I'll just mention this quickly in passing. You have relevance, new evidence of unexpected and significant impacts on biodiversity, urgency, actual geographic coverage, and potential spread. And as I go through this rapidly, try to think whether or not gene editing or genome editing will fall under this criteria. And then 3 more, evidence of the absence or limited availability of tools to limit or mitigate, magnitude of actual and potential impact, and then magnitude of actual and potential impact to the productive sectors. So you have magnitude of actual and potential impact to humans, as well as to the productive sectors and economic well-being. SynBio, as a concern, served as the introduction that will bring into the fray genome editing. So genome editing doesn't come together as one term. It simply is subsumed, okay, by the terminology release of synthetic life, cell or genome into the environment. And that was first brought to fore in the substa number 14, okay, item 16, paragraph 4, which was articulated in Nairobi, Kenya in May of 2010. Okay, so synthetic biology was not even given a full formal term. It was simply said to be present, to be part of synthetic life. And then in the same line, they mentioned cell or genome. But synthetic biology has now been utilized as a standard term, and synthetic life apparently has gone into oblivion. In COP10 in Nagoya, Japan, decision 13, paragraph 4, the formal usage of synthetic biology actually came into light when the COP-MOP invited the parties, other governments, and relevant organizations to submit information on synthetic biology and geoengineering. So in 2010, we have not seen the term gene editing yet in any of the documents that have been produced by the COP. Okay, they said that if synthetic biology is going to be part of the consideration for the substa or the subsidiary body on scientific, technical, and technological advice, it should comply with the conditions that have been set in COP 9, item 29, paragraph 11 or 12. Okay. Although it said that even if it has not complied with these requirements or these criteria, it is best to apply the precautionary approach prior to the release of synthetic life, cell, or genome. Look at the terminology: synthetic life, cell, or genome. Not genome edited, but entire genomes. In COP 11 in Hyderabad, India, decision 11, paragraph 4, said that noting based on precautionary approach, the need to consider the potential positive and negative impacts of components, organisms, and products resulting from synthetic biology techniques, okay, must actually be taken into consideration, and that the decision, however, has not accepted that synthetic biology is going to be part of the new and emerging technology that has to be discussed formally in the COP. So at this point, it's as late as COP 11, synthetic biology and all its attendant technologies, the grab bag of technologies, has not yet been accepted as a new and emerging issue in the COP discussions. In COP 12 in Pyongyang, South Korea, okay, again, it has not yet established whether or not synthetic biology as well as its attendant technologies are going to be part of the new and emerging technologies. So paragraph 4 established an ad hoc technical experts group, another one of your alphabet soup, ATEG, to identify the similarities and differences between living modified organisms as defined by the Cartagena Protocol and organisms, components, and products of synthetic biology techniques to determine if LMOs derived from synthetic biology would fall under the scope of the Cartagena Protocol. And then the major directive is for the ad hoc technical experts group to work towards an operational definition of synthetic biology, comprising inclusion and exclusion criteria using all relevant information based on scientific and peer-reviewed studies. So that was the directive given to the ATEG, okay? And this time you'll see that again, synthetic life, cells, or genomes did not include specifically genome editing or gene editing in the terminologies currently in use up to COP 12. Okay, again, decision 12, paragraph item 24, establishing an ATEG. The operational definition is its major directive, so that hopefully by COP 13, the operational definition would already have been set up. Okay, in the said convention, the Cartagena Protocol on Biosafety, CPB, it noted that synthetic biology can take into account the provisions of the Cartagena Protocol. And so whatever has been written in the Cartagena Protocol may apply to living organisms resulting from synthetic biology. It takes note of the conclusion as a substa, okay, relevant to the CBD, but not necessarily containing enough information to consider synthetic biology as a new and emerging issue. So at this point, discussions on synthetic biology and all its attendant technology packages is not yet formalized in the CBD. It urges parties and invites other governments to take a precautionary approach, however, in accordance with the decision 1111. The UNEP CBD, let me just get this out of the way. The ATEG of the UNEP and CBD said that the synthetic biology describes the application of various techniques of modern biotechnology that exercise control in the design, synthesis, or redesign of new biological organisms, plants, devices, and genetic systems. So in this particular regard, it tries to consider genome editing as being included or subsumed by the terminology redesign of genetic systems at the organismal, cellular, or subcellular level. And said now that for the very first time, it came into fore that you have genome editing taken together in a single breath. Synthetic biology, according to this ATEG of 2015, is particularly but not exclusively associated with the chemical synthesis, or this is where gene editing comes in, alteration of genetic sequences and nucleic acids, genome editing techniques, and an engineering-based approach to the construction of living organisms, and so on and so on and so on and so forth. So for the very first time, In the ATEG of 2015, genome editing was taken in a single breath. Now remember, this is a recommendation by the ATEG. This is not a decision by the CBD, nor of the Cartagena Protocol on Biosafety meetings. So what happens next? Let me just see if I can get this out. It's not moving. Okay. So in Cancun, the next COP, following the 2015 ATEG declaration, the COP did not accept the definition, but simply acknowledged. Sorry. Am I okay? I'm sorry. Okay.
Please continue.
Okay, sorry. Okay, so in COP in Cancun, Mexico, the COP did not yet adopt the definition provided by the ATEG, but simply acknowledged the outcome of the work of this ad hoc experts group on synthetic biology on the operational definition is actually seen. Okay, and essentially it acknowledged that the design, redesign, and manufacture or modification of genetic materials, living organisms, and biological systems would be a useful starting point for the facilitation of scientific and technical deliberations under the convention and its protocols. And so when it acknowledged the work of the ad hoc without actually accepting this particular definition, operational definition of synthetic biology, it seems to explicitly accept that redesigning the genetic material is tantamount to editing. In which case now, finally, the gene editing or genome editing technologies would now be formally included in the discussions on synthetic biology. Okay, COPMOP in Mexico takes note of the conclusion of the ATEG on synthetic biology that living organisms developed through current application of synthetic biology or in the early stages of research development are similar to living modified organisms as defined in the Cartagena Protocol. In this particular regard, there seems to be no real need to actually come up with new guidance documents for such products of this genome editing technology. Notes that the general principles and methodologies for risk assessment under the Cartagena would be a good basis for risk assessment. And notes also that it is not clear, given the current state of knowledge in 2016, whether or not some organisms of synthetic biology in the early stages of research and development would fall under the Cartagena definition of living modified organisms. And therefore further notes that there are cases in which there may be no consensus on whether the result of synthetic biology application is living or not. And to be able to finally decide whether or not synthetic biology and all its attendant technologies is going to qualify as a new and emerging technology, the COP in Mexico decided to extend the ATEG with renewed membership. In Sharm el-Sheikh in Egypt, Decision 1419 agrees that broad and regular horizon scanning and so on and so forth would be a legitimate activity by the Cartagena Protocol on Biosafety as well as for the COP. Unfortunately, in Egypt, whether or not synthetic biology and all its attendant technology, which now included genome editing, has not yet been established as a new and emerging issue, and a lot of discussion actually ensued with respect to whether or not it can really qualify. And so the ATEG, which failed to resolve the issue of whether or not synthetic biology is going to be a new and emerging issue, the term of its office is actually extended to be able to resolve this particular question. This Decision 23.7, okay, in Substa, the COPMOP decision in Sharm el-Sheikh left the decision to Substa. It decides to defer considerations on the submission that synthetic biology should be classified as NEI or new and emerging issue. And so it passed on the responsibility to the 24th meeting, considering the advice provided by the ATEG on synthetic biology. And of course, Substa 24 has said that the discussions on synthetic biology cannot yet be resolved. And if you look at this particular quote, it says everything is still under discussion. So at the moment, it stands that the issue of synthetic biology is not yet determined as to whether it is a new and emerging issue or not. And then there are things in bracket, and decides not to require further analysis on whether synthetic biology is an emerging issue, while keeping the convention's work in synthetic biology under review, still in brackets, and recognizing that synthetic biology has not been determined to be or not to be, again in brackets, a new and emerging issue. But despite that, it still said that let us, however, continue doing our horizon scanning. So maybe sometime in the future, maybe in the actual COP that's going to happen sometime this year or early next year, depending on COVID, we actually be able to resolve whether or not synthetic biology is a new and emerging issue, indeed. Okay, so decision 24 L5 of the SABSTA in Montreal earlier this year decided to say it's okay to do horizon scanning, and we will support 1 to 2 intersessional periods of horizon scanning. Recommended that an ATEG for broad and regular horizon screening be actually constituted, and recommended that the executive secretary convene open-ended online forum to support the ATEG on horizon scanning. All this without having resolved the issue of whether or not synthetic biology and all its attendant technologies, including gene editing or genome editing, is going to be part of the agenda where a lot of discussions will actually take place. Gene editing under CBD and CPB. Gene editing has come up in the discussions under the Cartagena Protocol as a potential issue requiring further risk assessment guidance. Of course, everywhere that it is mentioned in the recommendations in COP-MOP, you always see it bracketed. Parties cannot agree on whether it should be a specific issue. Parties cannot agree on whether or not synthetic biology is a single issue or made up of several issues that would have to be dissected, deconvoluted, and evaluated separately. The ATEG in 2019 recalled the ATEG 27 report, which concluded that most living organisms already developed or currently under research and development fell under the definition of LMOs as per Cartagena Protocol. And ATEG 2019 agreed that this conclusion was still valid. Most living organisms said. And so if it's under synthetic biology, most of it would be classified under LMOs in the Cartagena Protocol. The current state of knowledge, however, is that given the rapid developments in the field, it may be possible that synthetic biology organisms developed in the future could fall outside the definition of living modified organisms, the protocol. And it's already set a condition that if such a situation arises, it was recognized that the relevant obligations in the convention would continue to apply, to which the usual questions that are asked would be which obligations the convention would continue to be applied. And one parting shot of ATEG 2019, which gives me some bit of discomfort, is that it stated and noted that the convention contains a definition of biotechnology, which is broader than the definition of modern biotechnology in the Cartagena Protocol. It was recognized that all parties to the convention have obligations with regard to biotechnology and living modified organisms, and that the Conference of Parties has adopted decisions with regard to organisms, components, and products of synthetic biology. And in my parenthetical statement, I would say this would include genome editing. So if it fails to comply, the way I read it is if it fails to comply with LMO as defined by the Cartagena, as well as modern biotechnology as defined again by the Cartagena, it would go back to the mother statement or mother definition of biotechnology, which it claimed is broader than the definition of modern biotechnology. And I went back to the original convention and it said that biotechnology is the use of any of the living organisms to actually come up with a product that is different from the original.
Rita, can you conclude, please?
Yes,
Thank
yes,
you.
yeah, that's my last slide. So thank you. And again, I would just like to flash the thing that this is a personal view, and it does not include the views of the Department of Science and Technology or the Philippine government. I just want to use this slide as a last section as a precaution that maybe for every innovation, there's always regulation and convention, international or otherwise, that may drive the innovation into oblivion. In this, I echo Don Mackenzie's sentiment that perhaps the regulation and any convention that we sign would have to be science-driven So thank you very much. Thank you for listening.
Thank you, Dr. Carino, for your excellent presentation. Yeah, especially it was very impressive. The history of gene editing related terminology. Yeah, it was very interesting. And then
with Dr.
yeah.
Jan,
Yeah.
okay, just one intervention.
Mhm.
With the permission of the co-chairs, we have a provision to have the 2 poll questions for which we have to have the response from the participants. And if you permit, then I will request Dilip to drop the questions on the screen. And the 15 seconds will be given to respond quickly by the participants. And these 2 questions results will be shown at the end of the webinar. This will be helpful to us. The question is, in your opinion, the most relevant thing to consider when assessing potential hazards in a new variety would be, you have to have a single choice, the technique that is used to produce the variety, the parents used to produce the variety, the resulting phenotype of the variety, and the compositional data. The second question is, which category of gene-edited plants is likely to trigger regulatory oversight? Again, you have to choose one. Plants containing foreign genes from any source, plants containing foreign genes from a different species, plants containing point mutation, or all gene editing plants. So you have the 10 seconds to respond to this question. Please go ahead. I hope you have responded. If so, then I again hand over to the co-chairs to conduct the panel discussion. Thank you, co-chairs. Thank you, participants.
Thank you, Dr. Yagi. Well, then we'll have panel discussion time. And for this, I would like to hand over this to my co-chair, Dr. Mitchell.
Yes, thank you very much. Could I remind people to mute when they're not speaking, please? We've had a couple of interruptions. So the next item on the agenda is a panel discussion. So in this panel discussion, we have experts from 3 countries in the Asia Pacific region. So from the Philippines, Malaysia, and India. So I'll introduce the panel members individually before they speak. So our first panel member is Dr. Abraham Manola, who is a professorial lecturer from the University of the Philippines. And he is a planning and policy specialist with work on biotechnology and the biosafety policy, government restructuring, and formulation of development plans. So he's currently a professorial lecturer, and he's also the executive secretary of the Biotechnology Coalition of the Philippines, a non-profit organization working for the safe and responsible use of modern biotechnology. And I believe Dr. Manola is going to share his screen to present some information about regulation of gene editing in the Philippines and in other countries in the Asia Pacific region. Thank you, Dr. Manola.
Thank you, Dr. Mitchell. Can you see my slide?
Yes, we can.
Yes.
Thank you.
Yes. Again, thank you. So I'll be presenting on the case study of gene editing policy in the Philippines. And I was also requested by Dr. Viva to present for other Southeast Asian countries. So, the title of my presentation is Regulatory Approach for Gene Editing in Agriculture in the Philippines. First, some important milestones for the Philippines. Actually, there were already discussions on gene editing, CRISPR-Cas9, and similar techniques, even as early as 2014, 2015. I was looking at my notes this morning. But the formal technical study that was constituted by our Philippine Department of Agriculture in order to study all of these concerns, issues, innovations, and opportunities on gene editing, particularly for plant and plant products, started in 2017. So there was a formal group commissioned to do this study in 2017. And in 2018, before the completion of this formal study, the group conducted technical briefings and consultations with different stakeholders before they completed their study. And their study was completed in 2018, and this was adopted by the Philippine Department of Agriculture. Then a year later, this was submitted to the National Committee on Biosafety of the Philippines. So what is the NCBP? This is the highest biosafety policy advisory body in the Philippines. So this is the highest national body on biosafety policy in the Philippines. And when this was submitted to the NCBP, they took it upon themselves to study the technical study itself, and they constituted their own technical working group. And in May 2020, they formally adopted the Department of Agriculture study recommendations, and they issued a resolution. So this is the NCBP resolution 001 of 2020. And dear colleagues, before I go to the next slide, I would just like to show to you this particular resolution. This is the last page to show that this is a high-level resolution. Why? We're talking of 7 cabinet-level secretaries. These are in other government with parliamentary form of government. These are your cabinet ministers. So these are 7 cabinet ministers or cabinet secretaries who signed on this resolution. This is how important this resolution is. So what is this resolution? This is a resolution on how products of new breeding techniques, or now called plant breeding innovations, should be approached in the Philippines. So let me read, please, these highlighted portions. It says here that they resolve that plant breeding innovations may be classified as GMO or non-GMO, depending on if they contain a novel combination of genetic materials obtained through the use of modern biotechnology. So this is in layman's term, if there is a foreign transgene in the final product, if there is a foreign transgene in the final product, then it is only then when it will be classified as a GMO. And if it is classified as a GMO, it will be regulated as a GMO under our current regulatory system. But if the PBI-derived product is classified as non-GM, then it will just be considered as a conventional product. So, and third resolution, it tasked our Department of Agriculture to now issue the implementing guidelines for this overarching policy guidance from the National Committee on Biosafety of the Philippines. So very important milestone for us. It also presented a decision 3, how we will be deciding if a product is to be regulated as a GMO or non-GMO. And I'll not go into the details, but there are 2 important question points. First, was it produced through modern biotech? If yes, was there a novel combination in the final product? So that should be, it should be in the final product, not just as a temporary or transient phase in the final. If the answers to these 2 questions are yes, then yes, only then can it be a GMO and will be regulated as a non-GMO, will be regulated as a GM product. Then you might ask me, so what is the status now? As I told you, there was this mandate given to the, there was this task given to the Department of Agriculture to draft the implementing guidelines to this overarching policy. And right now, our Secretary of Agriculture, Secretary William Dar, issued Special Order number 303 in April 2021, who formed a advisory group. And one of the mandate of the advisory group is precisely to formulate guidelines in the formulation of the NCBP resolution number 1. So right now, this group is drafting the implementing guidelines based on the NCBP resolution. But I would like to give you an update that this work by the advisory group is also being guided by development in other countries. And in particular, as pointed out by Peter and Don earlier during the presentations, there are many countries already who can serve as guiding posts on how we should regulate PBIs based on this policy that if there is no transgene in the final product, then it is not should not be considered as a GMO. And so now we are this technical advisory group is trying to develop this questionnaire on what would be considered in order to determine if there is a transgene in the final product. So there are other countries also being considered here. For example, the one showed by Don earlier by Nigeria. But so these are the other countries that are being considered: Argentina and the implementing body, Brazil, Chile, Colombia, Honduras, US, USDA. Regionally, it's their MI regulated policy now under their secure rule, and of course Japan, where their PBI products is being regulated by 2 agencies, the Environment Ministry and the Health, Labor and Welfare Ministry. So these are the questions. For example, are the information required voluntary or required? There's only one country that is voluntary, that's the US under its USDA. And the breeding method or the grouping of the breeding method, alternative method used, the description of the intended new characteristic, submission of a genetic construct or genetic map, molecular characterization of the sequence, possibility and evaluation of targets. That's only in Japan and Brazil. Information about methods used to discard the transient genetic material. A couple of countries are using this. So information on authorization by other countries. So a couple of countries are also requiring this. And usage of the organism. Is this for food or feed or also for cultivation? So again, the Philippines is considering all of this plus others, for example, in Nigeria, to come up with our own set of questionnaires to consider if there's a foreign transgene in the final product. So that is the Philippines. That's the status. And hopefully by this year, implementing guidelines from the Department of Agriculture can be issued by the by the Secretary William Dar. But how about in other Southeast Asian countries? So I'll just be presenting those that are in a relatively more advanced stage in their policy regulatory development. So in Thailand, this is under the ambit of its biotech, formed a technical biosafety committee to make initial recommendations on guidelines for plant breeding innovations. They came up with a zero draft, meaning it's not considered as a formal draft yet. It's a very preliminary draft on PBI guidelines, but it is only for food safety assessment. So they're not talking yet of environmental safety assessment. So it's limited yet on food safety assessment. And this was presented on July 9 in one of their webinar. There's no English version yet. There is no formal English version yet published. And there was a mention by one of the officials during that webinar that they expected this zero draft to have major revisions, and there was a pronouncement that they will most probably adopt the Japanese regulatory framework, the one I showed you earlier. In Indonesia, still there are advanced discussions, but there are no formal policies that have been yet developed. And they said that the policy on gene editing will emanate from their National Biosafety Committee for Genetically Engineered Products. And government has organized several gene editing webinars and workshops. And the most recent important one was on January 2020, which involved the major stakeholders. There was a statement that the initial position in Indonesia that PBI products developed through SDN1 shall be categorized as non-GM and therefore exempt from GMO regulation. So this is the most concrete statement so far from the emerging Indonesian policy. And for Vietnam, they already started discussions on PBI since 2 years ago by way of a scientific outreach for public awareness. And so far, they have conducted 3 scientific workshops. Again, they have no draft guidelines yet, but VAAS, the Vietnam Academy of Agricultural Sciences, have initiated discussion on ongoing R&D developments because there are also PBI products being developed in advance to the design phase. So they are similar to the Philippines. They are discussed when we had our initial discussions in 2017. There are now also discussions on the state of the art of their R&D, the capacities of their local R&D institutions, and global and regional policy developments. The state of the art in regulatory development globally. And finally, among the 4 countries, except for the Philippines, among the 4 countries, Singapore is more advanced. So they already have a draft policy called Regulatory Approach for Food Derived from Genome-Edited Crops. But again, you will see here, although there's already a draft policy, again, it's only limited to regulatory approach for food derived from genome-edited crops. This has been developed by the Singapore Food Agency. They have already conducted public consultation with a second round of public consultation targeted in the near future. The SFA proposes a risk-based product trigger approach for foods derived from genome-edited crops. So this is the decision tree. But the last slide will show this table. So the main question is the genome edited crops containing foreign DNA? Then it would require pre-market safety assessment by the SFA and the GMAC. And it will require final approval from the SFA, and it should be identified as such. Otherwise, if it does not contain a foreign DNA in the final product, this food product, it does not require pre-market safety approval by the SFA, and it will not be identified as a GMO. So with that, I think that's my last slide. So thank you.
Thank you very much, Dr. Malolo, for an interesting review of the different regulatory statuses of gene-edited crops in the countries in Southeast Asia. If I could move on to our next speaker. If people have questions on that talk, please could they put them into the chat and we'll go through them all at the end. So our next speaker is from Malaysia. So we have Dr. Kody Isparan Kandasamy. So he currently works as the Group Science Advisor for the Asia Plantation Capital Berhad, which is a multinational specializing in sustainable commercial agriculture products, including forest plantations. He also serves on Malaysia's Genetic Modification Advisory Committee and the Science Ministry's R&D Grant Evaluation Panel, representing the industry. So please, could you give us our views on gene editing in Malaysia? Thank you.
Thank you, Heidi. Good afternoon, good morning. Inviting me to participate. Can I be heard? Is my voice clear?
Yes, we can hear you. Thank you.
Right. Okay. There was a remark saying internet unstable, so I'm not sure it's my end or somewhere else. I hope everybody can hear me now. In the interest of time, and I won't go. I'll be brief. As we know, the previous speakers, Donald, Peter, Frida, and Manolo has covered very much of the history of regulation, the evolution of plant breeding technology and GM. And I must say that we are not as advanced as the Philippines, where they have already come up with a resolution. We have not done that yet. And let me just describe through the Malaysian position. Malaysia's position is to support and encourage basic research to advance the understanding, the link between genes and diseases, especially for human and plants, and genes and productivity, particularly for plants. But saying that our major industrial crop is oil palm, and then in a larger scale, the commercial crops are paddy. The rest are basically smallholder farms. Yeah. So any technology development which will benefit the smallholder farms, which will be very much welcomed and supported. Now, of course, there's always a fear of off targets. That's the reason why we need solid scientific data to support the regulators in regulating this technology. As far as Malaysia is concerned, gene editing is still the definition of modern biotechnology is very similar to the Cartagena Protocol on biosafety. As such, gene editing is either it's unfortunate or fortunate, depending on who's the stakeholder. It's regulated under the Biosafety Act 2002. So we have the Act, as what Peter said earlier, the government has the Act. So the regulator just enforces the Act, and we do the regulating. And I'm part of the regulating organization. Although I represent the industry, trying hard to facilitate the industry, but regulation is part and parcel of what the law requires. So the law covers all research activities and resulting product that arises from modern biotechnology. So, as I said, gene editing regulated as it would be like any other genetically modified organisms or products. Well, the Act recognizes the immense potential, but it also recognizes the possible risk associated, thus the need to regulate. Yeah. So that's and as agriculture in Malaysia policies embraces modernization to address food security, climate change, etc., it is important to have an enabling legislation in place to build confidence for both researchers and public. Yeah, researchers on the end, they would like their product out in the market as soon as possible, but the public are generally very cautious. It's the disconnect in communication between the two, where the law comes and makes sure that it's satisfying for both. Therefore, as the law, the Biosafety Act 2007 allows for possible exemptions. Yeah, so there are discussions, but as I said earlier, we are not as advanced as the Philippines, where there is already a policy statement, or as advanced as Singapore, but Singapore it's easier because they don't do much cultivation, so it's more for food. Yeah, so food derived from anything like GM organisms or gene-edited organisms, whereas ours, we do cultivate, and so we have our process will be a little more tedious. But this discussion and making exemptions inevitably means takes time, as various deliberation discussions have to take place amongst the many stakeholders. Yeah, so the researchers, the government, the public consultation is a must in whenever there is such exemption to an existing law. So up to date, although we wish so much, up to date, we do not have any gene edited or GM plants gone from lab to land. And as a genetic engineer myself and as a commercial planter, I would love to see, although I love to see the plants, GM plants or gene edited plants out in the field in Malaysia, but it has not happened yet. I hope it happens very soon. It will be a fantastic test case for the regulators in Malaysia to look at how we regulate a plant, taking it from the lab to the land. Except palm oil and paddy. Paddy is also very much smallholder driven, but palm oil is the industrial crop. I won't mention rubber because rubber is not food, and it's into. But smallholders will benefit. A lot of the vegetable productions are smallholders. Yeah, daily consumption products are smallholders, and so these technologies will definitely benefit the smallholders. Okay, that's much I have at this point, and I'll be willing to answer any questions if there is any. Thank you.
Oh, thank you very much for those insights. Now, our final speaker from the panel is Dr. Vibha Ahuja. So Vibha is the Chief General Manager of the Biotech Consortium of India Limited. So she's an expert on biosafety and regulatory aspects, particularly with reference to genetically modified organisms, having more than 25 years of experience in the field. So Vibha is going to talk about regulation of gene-edited crops in India. Thank you very much, Vibha.
Thank you, Heidi, and thank you, Dr. Jang, for giving me this opportunity to share the Indian situation. As we are already over time, I will just quickly inform the audience about what's going on in India. In India, all the genetically modified organisms, engineered organisms are regulated as per our rules, which were laid in way back in 1989. And as at that time, all the things were evolving. We have a definition of genetic engineering in our rules 1989, which is very broad based. And also there is also the definition is broad based, and also there is a provision in the rules that all new genetic technologies will also be subjected to regulation under these rules. So since these rules were, as soon as the discussion on gene editing started, it implied that this would be covered under the rules 1989. However, our Department of Biotechnology, which is one of the regulatory agencies, the Ministry of Environment and Forest is the apex agency supported by the Department of Biotechnology and the state governments in the monitoring role. So the Department of Microbiology and has taken up initiative and drafted the guidelines for gene editing in December. They released it in January 2020. But these guidelines encompassed everything, the plants, animals, microorganisms, everything. So there were a lot of consultations in the country, including by our National Academy of Agriculture Sciences, which recommended a very nice framework particularly for the gene editing in plants. Now, this is the framework which has been used by the Department of Biotechnology. It is almost similar to the new guidelines. The new guidelines have been made and which are under discussion by the regulatory agency. But as far as we know the details of the guidelines. The key factors is that SDN1 and SDN2 categories of genome edited technologies, which do not involve or carry any exogenous DNA and are comparable to the naturally occurring variances, will be exempted from the rules 1989. Although this process will take a little more time because the apex committee, which is Genetic Engineering Appraisal Committee, has to approve it, and then there might have to be a legal amendment, kind of an amendment issued to the rules 1989. But we are still waiting for these decision by the apex committee. Although the guidelines also indicate that the initial activities, research activities, which will be undertaken under the contained conditions, laboratory, greenhouse, till the time you confirm that that the product is transgene free, it would be regulated under the guidelines. But subsequently, once it has been demonstrated in SDN1 and SDN2 that we do not have any transgene, then it would be subjected to the regular laws for the testing of the conventionally bred varieties. The products of SDN3, which will be classified as the genetically modified organisms, and they will be treated in the same rules as Rules 1989. So this is the broad approach which is in the pipeline, and but this is still under consideration of the apex. We are hoping that it is.
Phoebe, Phoebe, you're muted. Please, could you turn your microphone back on?
Sorry. I was just saying that we are waiting for the final decision. However, we are hoping that this would be fast because we are seeing in the several other documents, like our recently released National Biotechnology Development Strategy, where it has been indicated that several mission programs are going to start, centers of excellence on gene editing. So there's a lot of emphasis on the use of this technology in agriculture by all our apex bodies like Department of Biotechnology, Indian Council of Agriculture Research, and so on. But definitely the regulations will play a key role in bringing the fruits of this research to the use by the farmers. And so we are just waiting for these guidelines to come out. Thank you. This is all I have to say.
Thank you very much for that brief presentation. Now, I realize we are over time. Can I pass back to the organisers to see whether we want to continue with questions or whether we think we should wrap up given the time?
Yeah, maybe a few questions, 2 to 3 questions can be taken up. Dr. Tyagi, you can guide us on that.
Yes, I agree fully because we are over time, and maybe 2 to 3 important questions can be taken up, which are relevant to this webinar, because several questions have been asked about the IPR and all that. So in the next webinar will be for that, but the relevant question 2 to 3 may be taken up by the chairs.
Okay, and I know Dr. Zhang, you had some questions that you were hoping to ask. Would you like to go to those now?
Yes, thank you. Thank you very much for giving me the opportunity. Well, I'd like to bring up a very basic question that is about the definition of the products gene edited. Yeah, well, there are many countries who are parties to Cartagena Protocol on biosafety. Well, Korea is one of them. And then even if there are parties to the protocol together, but the way they interpret the definition of LMO, GMO sometimes seem to be different. For instance, a certain country define the products, gene edited products like this. When gene product does not contain extracellularly made material, then it did not fall under the definition of LMOs by the protocol. By the way, according to the interpretation of the definition of LMO in the protocol, well, I'm looking at the explanatory guide to the Cartagena Protocol on biosafety. It is written like this. Yeah, interpretation. The word that I think most important is the word genetic material, because the living modified organisms contains a novel combination of genetic material produced by modern biotechnology. Well, then the protocol explanatory guide, according to it, it says, even if a novel combination of genetic material did not result in observable change in, for example, the phenotype or appearance and behavior of an organism, the combination would still be novel. The novelty of combination could arise through the presence of a novel form of a functional unit of heredity, resulting from a change that modifies the unit by altering, inserting, or deleting one or more nucleotides within the unit. According to this, any change to the nucleotides, whatever small, it could be interpreted as LMO by the protocol. Well, in this aspect, I would like to ask this question, opinions. From, I think all the 3 speakers' presentation are related to this. Yeah, anyone would be okay with my question? Yeah.
May I?
Yeah, sure.
Well, the way I see it is, if it's not part of the original document, and it's just an interpretation
Yeah, right.
by some by even a technical group, then I think it should be then I think it should be still open to negotiations.
Mhm,
So I would I would argue
mhm,
that because there are already so many single nucleotide polymorphisms, for example, in any organism, it might be very, very difficult, if not impossible, to actually identify which among the gene edited products would actually be
mhm,
considered LMO.
mhm.
So that's my take on it. So if it's not part of the original document, I will take it as still open to other interpretations and can be negotiated in the COP.
Mhm.
Thank you.
Yeah, thank you.
Heidi, one question I have picked up, which we have been asked during the registration. It is a common to everybody's interest. And I would like Don, if he's there, he can respond, or any other speaker. How do we achieve policy harmonization across the border for gene editing? What is the command? It is a very general question, but it is the interest of everyone and curiosity everybody has here. Can Don respond to?
Yeah. Thank you. That's not an easy question, but I think one of the ways is actually through having webinars and meetings such as this one, where we get the relevant stakeholders together and interested parties, and we talk.
May I request to mute your speaker, please? Madappa Mali, can you mute? Thank you. Don, please continue.
Yeah, as I was saying, I think one of the ways to try and achieve harmonization is through these kinds of dialogues that we're having here today. It's a good first step because it helps to sort of broaden the understanding of the issues amongst all of the relevant players in that. And I guess at the end of the day, governments of different economies need to be of the same motivations. They have to realize and appreciate sort of where they want to go and how they want to deal with the technology, and whether they want to sort of continue to take a very precautionary approach or a different path. And I think that's one of the things that we're hoping with this particular suite of technologies is that economies do take a different path than maybe they chose with the first round of sort of genetic transformation technology.
Thank you.
Yeah.
Could I ask a slight follow-up question to that one to the speakers as to what do you think the worst outcome for gene-edited crops is? Is it the continued uncertainty about their regulation, this kind of patchwork that we've got of the moment of some countries considering them as GMOs and some not? Or is it everybody regulating as GMOs? What is the worst?
In my view, Heidi, I think it's uncertainty is probably the worst. And I don't think it matters whether you're an academic researcher working on a project or whether you're a large corporation in that. Uncertainty is the hardest things to deal with. If you know what you've got to do, you can make a plan to deal with it. And when you don't know, it makes it really almost impossible. And trade and everything comes into it as well. Yeah.
Thank you. Did you want to add to that?
I was going to, so I wouldn't proffer, as a regulator, I wouldn't proffer an opinion about what the worst outcome was. I thought what Don said then was very, very appealing. I would say that even with GMOs, there's something of an analogy already existing in that the US regulation, for example, is quite different to the European regulation and their major trading partners. So that uncertainty probably exists there to some degree. I had another thought which I've forgotten, so I'll stop talking.
Okay, thank you. Dr. Jiang, did you have other questions that you wanted to pose?
Well, I think we have many questions which are in the chat box. We can choose some of them. Can you, Dr. Heidi, did you read some of them? I'm now just looking at.
Can I jump in straight away? One of the questions I saw early was about whether gene drive organisms would be considered as GMOs. And I think from the Australian perspective, and I suspect for just about every process trigger-based system in the world, they will be, because they are qualitatively transgenic organisms, in that unlike the genome editing we've been speaking of in these webinars where small changes might be made, the whole, even though you talk about CRISPR gene drives, it's the addition of the whole CRISPR gene plus the gene of interest. So they're clearly GMOs, transgenics, according, to the, they don't fall in the same basket as genome editing that we've been discussing here, in my opinion.
Thank you.
May I make a comment on that one?
Yes.
Yeah, with respect to the gene drive and genome editing and CRISPR, they, if you'll notice in the discussions in the Cartagena and in the CPB, they always lump them together. They don't want to deconvolute them. So it all goes into the same basket. And so that's why there is a great discrepancy with respect to the opinion about synthetic biology. So I think that is the problem with it. If you lump gene drive with genome editing with all the other tools of synthetic biology, then you're going to encounter a lot of problems, because they're not the same. They should be assessed based on their own merits, and they should not be lumped in a single pile. But it's very, very seldom that you're going to see them discussed in separate sections. They're all under synthetic biology. And now they're even introducing now the DSI into the same pile. So that's going to be a lot of a problem.
Okay, thank you. One of the questions in the chat is about, I think it's basically to Viba. In the case of the Indian scenario, where will CRISPR-Cas derived products be grouped under? If you see the end product, it is as good as derived from mutation breeding. These gene edited lines should be considered under group. So is that a question that you can respond? Yes. Thank you.
Yeah, very much, like this is the point that has been made was the main consideration during the discussions, that the products of the SDN1 and SDN2 are similar to that of conventional breeding. But however, it has been discussed, and the new guidelines suggest that the preliminary work that you do in the recombinant DNA work in the laboratories should be reported to your institutional biosafety committees. And once you have reported that this is there is no transgene and this is similar, so it would be treated in the same way as the conventionally bred varieties and subjected to the same rules. So this is the thinking behind the new regulations. I hope I have understood and responded clearly. Thank you.
Thank you. Thank you.
There's always a contention. There's always a contention that regulation is really difficult and makes everybody's life difficult. But it doesn't have to be that way. It can be regulated, and it can be seamless, and it can be made simple. It's how we handle regulation, I think. Whenever we talk about regulation, everybody gets very frightened. And being a regulator myself, I think the drive will be to make it, look at proper scientific data and make it as easy as possible, both for the researcher and the end user. I think we have to make regulation easier, not so frightened, frightened everybody, and
Thank you. One of the other questions that came in before the seminar was about whether genome edited plants and genetically modified plants would be treated by the public with suspicion. So do people here think that genome edited plants are more acceptable to the public? Would anyone like to jump in?
It's a lack of communication, I think, in Malaysia. I can only talk in the scenario in Malaysia. It's a lack of communication. The public is not so well informed, who's so terrified the moment they hear that it's anything to do with genome or genetic, and so it's a matter of communication, I think, and creating awareness supported by good science, and that so it will make everything easy if that's communicated. A lot of the hurdle is the lack of communication, I think.
Dr. Heidi?
Yes.
Well, I have a question about the Australian regulatory system. Well, this can go to Dr. Peter Tiggerson. Well, you told us that SDN1 can be excluded, but from the SDN2, it's impossible to be excluded. So, but in some cases, there are examples that SDN2 also can be excluded. What is the difference from your decision and other countries? What do you think about that?
So probably the best way for me to answer that question about, which I might reframe as, why is the current change or the most recent change to the Australian regulation limited the exclusion of a genome edited organism to site-directed nuclease without the addition of a template that we all call SDN1, was for a mixture of scientific and policy reasons. So the review which resulted in that change was a technical review initiated by the gene technology regulator, and not a policy initiated review. And in the conduct of that review, it was felt that to move beyond that situation, where you had a very clear analogy with single base pair changes with traditional mutagenesis techniques, to move beyond that into the SDN2 in the context of the broad definitions would be a policy question rather than something for the regulator to propose on her own initiative. And so that was taken up in a sense, if you recall that slide that I skipped over very quickly, in what we might call the big P policy review, with the recommendation indicating that there might be further updates to definitions which might include further exclusions, but that was as far as the technical review was able to take it, and I simply don't know if there will be changes from the big P policy review. So that's probably not a very satisfactory answer to your question, but that's my understanding of how we arrived at that, is that's what was proposed and done at the time. If someone wanted to read about this, there were actually 4 options proposed for the regulator's review. And in terms of that being taken forward, there were reasons given for why that option was chosen. And I won't attempt to recount those. I think that information is still on our website. But to sum up, that's the kind of pragmatic policy decision that's been taken at this point. And as I said, you watch this space to see if there are further changes to the Australian regulation.
Thank you for your kind answer.
I'm conscious of time now. It's, I believe we're 30 minutes over time. Do people have any pressing questions?
Can I give it? Can you allow me to speak a little bit?
Yeah, you can.
It's from Professor Imdadul Haq from Bangladesh.
Yeah.
So thank you very much for inviting us. And I'm sitting here from the very beginning, and I listened all the presentations. It was very nice and very informative. So just my concern and some confusions, like we started biosafety regulations, and when after the emergence of the CPB, it was in 2000. Now we are into 2021, so almost 21 years passed. But still, we have a lot of confusions, even among the regulators and also scientists. Like, for example, vitivinjal. This product, they started in 3 countries, Bangladesh, Philippines, and India. So it was started in the year 2004 or 2005. And Bangladesh already released in 2013, and it has been cultivated commercially in Bangladesh.
Dr. Haq, may I intervene for a second? If you have any question, please pose that question, because we are already running over the time.
Yeah, no, just
so we are respecting everybody's time.
Just 1 or 2 minutes. So what I am telling about, just the Philippines, they released after 7 or 8 years. India is still waiting. So my concern about the new technologies like genome editing, if we actually want to do something, we need to sit together, like the developer, regulators, and scientists. If we don't like sit in the same table, it will take even like 20-25 years to develop a consensus documents on the genome editing also. So my concern is we are always confusing with the new technology. It should not be, because Don mentioned that we have long gaps from lab to land. So that must be reduced. Otherwise, we cannot get any benefit out of the new technology. Thank you very much.
Thank you, Imdadul Haq, Dr. Haq. Thank you. Well,
yeah,
Dr. Mitchell, I think we had better turn our microphones to organizing team. What do you think of that?
Co-chairs, I would request you to give your closing remarks. And then I would like to formally thanks all. So before that, your closing remarks, if you have anything to say, final word.
Yeah, well, if I take first, I think we have a very excellent time on genetic engineering in continuation with the first webinar on 2 weeks ago. Still, I think we are rich in terms of time and opportunities, because we have another webinar 2 weeks after. And many questions still be waiting for being answered. But in the next webinar or otherwise, then through the printing materials, I think we can deal with those. I'd like to ask it to the organizing team. And this webinar has contributed to to better understanding the context in countries on gene editing, especially in terms of regulatory policy and practical issues. And I'd like to believe it will end up helping us harmonizing in the field of various aspects of biotechnology for the best of all. Thank you very much. And
may I invite Heidi?
Thank you. I would like to echo Dr. Zhang's comments. I think one of the important things about harmonization is, first of all, understanding where everybody is at and what their thinking is and how they got to where they are with their regulation. And then you can build on that to find synergies between different countries and then build on those to try and achieve harmonization. So I think this is a great first step in that path to trying to appropriately regulate or not gene edited crops. And I think this will provide us a lot of material for the written work. So thank you to all the speakers, the formal speakers and the panelists.
Thank you, Heidi. Thank you, Jan. May I request Dilip to show the results of the poll questions quickly, please? Oh, that's good. So the results are before you. And the first question, 41% has answered the resulting phenotype of the new variety. And the second question, 35% has mentioned that plants containing foreign genes from a different species. So this category may trigger the regulatory oversight, but many people are differing 25% in different way for any source. So I think it's a mixed opinion, and it is reflected in the general society also, in the research scientists also, and the other regulators also. So it will be helpful. Thank you, participants, for participating in this poll question. And now, with the permission of the co-chairs, I have one formal vote of thanks. And let me first of all thank the co-chairs. For the paucity of time, I will not name each and every one. So my sincere thanks to the co-chair for efficiently conducting this webinar. To the speakers who have taken a lot of words to make the presentation and responding to the queries or clarification from the participants, and the panelists who made this discussion very vibrant. And I would like to thank the organizers also, particularly the Korea Biosafety Clearinghouse. The director is here, Dr. Homing Jang is here. And the PCIL, Dr. Vibha also working as a panelist. And in fact, to tell all you, that Vibha and her team has been doing a lot of efforts and to organize all this series of webinar in this project. So thank you, Dr. Vibha and your team. And I would like to thank my colleague in Paris Secretariat who has done a lot of background work. Thank you so much, all the participants, for making it very successful, because a lot of participants are about 390 I see on the screen still. And I invite you to all for the webinar 3. As I mentioned, we are having a series of webinars. That webinar 3 will be on enabling policies for enabling policy for genome editing in agriculture. And many of the questions what have been raised or not answered, or you could not get the opportunity, maybe about the regulatory policies, you will get the opportunity in webinar 3. So all you are requested to stay tuned till 18th of August and the same time, and you can use the same link. And till then, thank you and bye-bye. We stop here with the permission of the co-chairs this webinar. Thank you, co-chairs.
Thank you.
Thank you.
Thank you.
Thank you.
Bye.
Bye.
Okay, bye-bye.